officer getting crammed in. Who knows what's going to happen by the end? But I want to talk about interdisciplinary legacy of Egenis, which I think is productive interdisciplinary collaborations of scholars across the divide of different sciences and the humanistic study thereof, and especially, as Stefan just mentioned, in the context of large projects. What I want to do is honor that legacy with reference to my own research in a similar vein, share with you some features in terms of both large projects and individual collaborations. So I'm going to talk about the big project I'm currently involved in. Some of the people who are here are also taking part in that, which is on teleology, and then talk about some local activities at the Minnesota Center for Philosophy of Science that I'm engaged in and direct, and how that cultivates interdisciplinary intellectual community, and then do some examples of my own collaborative efforts with scientists. So let's go ahead and start here with the large-scale interdisciplinary project to interrogate one of the most controversial aspects of life science, teleology. This audience knows a lot about teleology. It's certainly very old. It's been around for reasoning about the life sciences from before there even were life sciences. There's been a lot of diverse and contradictory interpretations. But it continues to be salient today. It has never gone away, and yet it has become very charged because of certain developments in biological theorizing, perhaps most notably evolution and mechanism. It has come in for its own sort of interesting treatment. Some of you will be familiar with these quotations. I've nicknamed this teleophobia because if you talk teleology, you could actually make many biologists blanch. And yet, as David Haig has recently said, I was repeatedly warned against teleological thinking by lecturers who spoke of the heart as a pump for the circulation of blood and of RNAs as messengers for the translation of proteins. Concepts of purpose and function shape the practice of biology. This is the natural way to think about living things, but the overt language of teleology is censored. So what I see out of this and got interested in is that you've got the situation where the descriptive language that imputes purposiveness at different times and in different ways is used, but it's worried about that that's problematic and possibly an error. And yet we're also seeing a growing recognition that a number of biological phenomena might demand new conceptual frameworks that have been neglected precisely because people were scared to come too close to them for fear of teleology. So could we build a research program? Given that purposive phenomena remain central, can we recognize where we are and potentially move forward, investigating these phenomena scientifically, simultaneously be careful and rigorous about the use of our descriptive language, and not merely use it, say, as a heuristic convenience? What if a research program was organized to come at these questions from multiple conceptual, theoretical, and empirical angles simultaneously? A systematic interdisciplinary conversation that brings together a wide range of conceptual resources. We're talking about purpose in the context of model building, experimentation, and theorizing in biological investigation. But what I've just done is given you a pitch for what we're doing. That's the project. This is the project website. The whole idea was to basically see, can we put this on a kind of progressive footing to go forward rather than simply get stuck in definitional debates and worries about the nature of teleology. So this is the project, agency, directionality, and function. It's a cohort program to address this demand through a novel interdisciplinary large-scale program that combines philosophers, theoreticians, and experimentalists. So importantly, the interdisciplinarity is interdisciplinary with respect to the sciences themselves, in the sense of having some mathematicians, having some people who are experimental biologists, some paleontologists, but also philosophers as well in that mix. To articulate more precise concepts related to function and purpose, develop innovative formal models of agency, operationalize notions of goal-directedness for accurate measurement, and trial and implement methods and platforms to detect and manipulate directionality in living systems. So the whole thing is got a kind of basic architecture, which you can see here. And in that structure, there are 24 subawards around the world. Those 24 subawards are collected together into clusters, 7 different clusters who each have what I like to call an intellectual shepherd, who is a philosopher, who has a specialization in the theme of that cluster, and the clusters are organized around key concepts, modeling practices, and distinctive phenomena at diverse temporal and spatial scales. You can also see there's a scientific board of advisors who are there to actually evaluate the research as it's ongoing, to give perspective on whether or not it's making the progress we had intended for it to make. This shows you those clusters represented in a different way in the space of agency, function, and directionality, sort of showing you that the projects kind of touch on different themes. They don't all touch on all the themes simultaneously. There's a kind of spreading out in the space with respect to what they are doing. One way I like to describe what we were attempting in this project was to move from metaphysics to modeling, a kind of changing of the question. Instead of asking what is teleology, kind of metaphysical question, instead ask how do biologists study things like agency, directionality, and function, for what aims, and do their experimental strategies and modeling practices serve these aims. And what that can do, and this is part of what Max Trezau and I argued, is it can move us beyond being scared about teleology, so scared that we had to use a new term, teleonomy, which turned out to just be confusing because everybody just meant the thing I want to study that I don't want associated with teleology. And in fact, I think it was a conceptual regress in the 20th century. Let's see, this is, here we go. Okay, so, the one last thing I was going to say about this previous slide, which it's going to let me come back, is that when Asa Gray was commenting on Darwin's work, he said, usefulness and purpose come to the front again as working principles. I think this is kind of a nice slogan for what we're trying to do, which is actually trying to make these things workable principles in the science. I think that's where a lot of payoff is. So this is one way I've tried to articulate it since the project began, that you first embrace teleology, you don't run away from it, and you directly try and examine purposive seeming phenomena and conceptualize living systems teleologically, figure out the value of that, then fragment. I think you need to adopt a pluralist stance. Teleology is like other biological concepts, means many things, right? Gene, homology, species, we're familiar with this. Treat teleology in the same way. Stop trying to say what it really is. And of course, that fragmentation is a good epistemological strategy that's shown up historically. Then characterize it, track distinct patterns of additional concept use, which are always in play, introduce distinctions, attend to different scales, and then apply that in modeling strategies, data gathering practices, operationalizing concepts, uncovering empirical signatures, and selecting abstractions, approximations, and idealizations for modeling. So that you walk through this kind of a sequence with a specific intent to try and come out on the backside of it with something that is changing the science. And importantly, as a research program, you have to do it over and over and over again. It's not a one-time thing that you say, okay, now we've finished that stage and we don't have to go back to it. It's a constant cycle. If you're curious, come see my face. Now I'm going to move on to my second part, which is the local activities at the University of Minnesota through the Minnesota Center for Philosophy of Science. So the Minnesota Center for Philosophy of Science is similar to Egenis in the sense of being a research center that collects together faculty from different units across the university around a variety of activities, some more regular, maybe some less regular, larger conference type activities. And it has a historical legacy of interdisciplinarity. Some of you may know this history. In 1923, Herbert Feigl began his doctoral studies under the supervision of Moritz Schlick. At the University of Vienna, a philosopher studying how early 20th century developments in mathematics and physical science were transforming our understanding of what knowledge is. Within 12 months, partly at the prompting of Feigl and other students, an interdisciplinary discussion group led by Schlick coalesced on Thursday evenings. It became known as the Vienna Circle. At the time, it was also commonly called the Schlick Circle. The reason why this is important is because it bears on the origins of the Minnesota Center for Philosophy of Science, because Feigl left Europe in 1931, taking a position at the University of Iowa, where he stayed for 9 years before coming to the University of Minnesota, and then started the Minnesota Center for Philosophy of Science on the model of those Vienna Circle meetings, with similar motivations. The aim of this reflective enterprise was the clarification of the basic assumptions, concepts, and methods of the sciences. Now, the reason why I go back to this history and thinking about the present is because it helps us understand a lot about what it means to have an intellectual community like has been here at Egenis and elsewhere. The first thing is to recognize how not to do it, right? And that's not by getting everybody who agrees on everything together. The Vienna Circle actually is a really nice example of this. They really actually strongly disagreed about most of the things they talked about. They're not always remembered that way, but it's clearly documented historically now. And also it's not about the standard academic structures that we all inhabit in terms of professional societies and or formal membership. It's more about informal networking, disciplinary boundary crossing, and that was also a hallmark of the Vienna Circle. When you think about how to build it, right, here, some of the recommendations are not, as Stefan highlighted earlier, rocket science. They're things that are pretty easy to do. One is a shared orientation. Of course, the Vienna Circle takes seriously the latest developments in logic, mathematics, and sciences. But notice how open-ended in some ways that is. Take seriously. Right? You can have a lot of disagreement under a take seriously umbrella. Regularity in timing and format. We are creatures of habit. This makes a huge difference. Relatively flat social structure, where diverse participants can all contribute and not feel like some voices are more important than others. And multidisciplinary composition, so that you don't have everybody coming from that same viewpoint of a particular disciplinary training. And that changes over time because the group is organic and sees people come and go as a natural part of academic life. And that's what we try and do at the center in Minnesota through our different interest groups that collect together people under these different sorts of topics. Biology, early modern, foundations as logic and math. The center discussion group is the original one that Feigl started, and that focuses on general philosophy of science. We also do conferences and workshops, a colloquia series, all things that articulate this structure. And we do things that are very straightforward. For example, when a weekly meeting of the biology interest group happens, everybody introduces themselves every week, even if everybody in the room knows who's there, because you never know when somebody new is there. And when the new person comes, they don't know who's in the room. And that's a way of being open to incorporating new individuals. Now, part of intellectual community is intellectual hospitality, right? We want to be in community with people who we like to spend time with, we'd like to have meals with, we'd like to share a drink with. And this recent book by Paula Rance Cohen has actually been really helpful for me in thinking about some of these things. Intellectual hospitality is a crucial ingredient for interdisciplinary communities, especially when you're going to disagree. Because if you're going to disagree, you need to be able to go have a beer afterwards. Anyway, right? But hospitality that involves conversation also requires time. And as Cohen has highlighted, faculty are focused on their own research and don't want to take time away from busy schedules to trek a block or two. One might even say leave the building. But it's important to have a community of scholars who gather together and talk. If faculty do not practice conversation of the best sort among themselves, it is unlikely that they can bring these skills to bear in the classroom and elsewhere. And the analogy that I give is that you have to practice interdisciplinarity. If you don't practice, you won't do well in the game. just as you wouldn't expect a football team to go out on a World Cup pitch and do well, having seen each other for the first time, right? So too, research teams and people are going to do much better if they've actually practiced before they play, right? So some lessons that I think we can take away from this: there's not a good or ideal time to undertake intellectual hospitality. Other things are always going to seem more urgent. Just recognize it. If it's important to you, you'll do it anyway. And I mean that also not just in the sense of academic busyness, but also in social and political times, right? Because the Vienna Circle was not happening under lovely conditions in Vienna, okay? And so too, our own times have their own complexities. Focus on topics and themes of interest to those who are involved, right? Read one another's work. One way to do that. Do it regularly. Encourage discussions to revolve around understanding claims and why they're being made, especially to identify standards of evaluation. This is really important in interdisciplinary context because people don't understand why something is an important claim, how it's justified, and all that has to be surfaced in a way so that people can understand it. Part of that is watching for the introduction of jargon and do vocabulary checks. I actually introduced in some of my workshops recently, some have seen this, actual tool, a high-tech popsicle stick with a red and a yellow side that you could raise up and say, I need people to pause or to stop because I just don't understand what you're talking about. And that's, I think, really important to have those sorts of abilities in the group. And then, of course, cultivate habits of active listening to discern patterns of assumption and styles of reasoning. Lots of us in academia are good talkers. Fewer of us are good listeners. But the groups that work really well oftentimes have very good listeners in them. And that's what makes them operate at a high level. So that was the second part. Now I want to turn to a few examples of my own collaborative efforts with biologists. This is kind of more in the vein of just sharing some of the ways I have worked closely with scientists and some of the things that have come out of that. I'll look at three recent examples: the living fossil concept, what is a trait, and the nature of mutation. So in collaboration with Scott Lidgard, who's at the Field Museum in Chicago, we wrote a paper rethinking living fossils and have developed it a little bit further over a couple papers. And in that, we were looking at the contested concept of living fossils, which many have argued should just be thrown out completely. Many of the paleontologists and evolutionary biologists have thought this. But we wanted to say, can we take seriously both its routine use across disparate research questions in biology? Because if you do literature searches on it, it's being used regularly and across many disciplines. And the worries about its misleading inferences in order to create new paths for productive research. And so as a part of that, we documented the different criteria that are often used to designate living fossils, recognizing that those criteria are multiple. We cataloged the complaints that are oftentimes leveled against that to try and make sense of this space, which was receiving this kind of criticism and yet was clearly not going away. One of the things that we noticed was a part-whole ambiguity that is something more generally applicable across the sciences, but was particularly relevant in the case of living fossils. All scientific practice involves using proxies, measurements of particular properties that stand in for an entity or phenomenon. Biologists often use subsets of characters and their differing states to discriminate among organisms or lineages, and that's extremely important in the case of living fossils because you don't get all the parts that you want from an extinct taxon. And so there's an ambiguity often between morphological and molecular parts of an organism and whole organisms or genomes of lineage or clade when they're being evaluated for stasis. And so you can have situations where the morphology looks like there's stasis, and then the underlying molecular analysis says, hey, molecular evolution is happening at a normal pace. Is it still a living fossil? And there's a part whole. Which part represents the whole? And that comes up in these contexts. This is maybe easiest to see visually for fossils themselves in the sense of paying attention to one particular feature. And if you do an analysis, say, on the tail fin length, as opposed to the pectoral fin length, as opposed to the eye width through a particular series of fossil layers for a lineage or a group of lineages, you can actually get extremely different signals from random walk and gradualism to stasis for the different characters. And again, does the part represent the whole? How do we talk about the lineage exhibiting stasis under these kinds of conditions, and does it make sense to do so? Another feature of this work was to recognize that what the concept of living fossils was doing was going beyond just categorization. A lot of the criticisms were about categorization. If I use my criteria, it doesn't fit. If I use another criterion, it does. But was a living fossil really about putting things into bins, so to speak? That is it really about whether it's inside or outside a particular criterion's boundaries? In fact, what we saw and discerned was that when you recognize that concepts play many roles in human cognition, generally and within scientific reasoning specifically, then we could recognize other roles like concepts playing a role in representing broad investigative domains. And we think actually that's a primary role for the living fossil concept, that it actually marks out a space of what needs to be worked on or explained in a given instance, especially with respect to morphological and molecular stability or persistence over long periods of time. And what we went on to do was to categorize and sort of catalog the different kinds of questions that are in that research program. I'm not going to read them to you right now, but the point is just to say that as a research program, it's got many things that are of interest. And some scientists are working on some of these questions and some scientists are working on others. And it's not surprising that you might end up with some clashes in how they conceptualize living fossils across some of these diverse research questions. Okay, my second example is from questions about what a trait is or what a character is. This is joint work with Gunther Wagner and James DeFrisco. We all know biologists frequently talk about characters, traits, and a variety of other terms to pick out aspects of organisms they want to study. There's a whole diversity of those things. Some of them are material parts, some of them are rates, some of them are activities that organisms display, some of them are quantitative traits. This is just a very small sample. And in response to this, one concern you might have, and one concern that Richard Lewontin had, was how are we to recognize the true characters of organisms rather than imposing upon them arbitrary divisions? No issue is of greater importance in the study of biology. And some of you will remember that this also came up in the famous Spandrels paper from 1978, although it was not the focus of their discussion. So our situation here, I think nicely captured by Alex Rosenberg and Karen Neander, that biology encompasses many different fields of specialization, and there will be no one-size-fits-all formula for taxonomizing biological traits. And yet the theoretical question of what constitutes a trait has not been systematically explored. That is, you can find some discussions here and there, but for the most part, people just start talking about traits as if they know what they are. Can we make progress here in collaboration with scientists in developing new theory in this domain? And partly this is an interesting thing because it's something that David Hull recognized many, many years ago. A place where philosophers could have been of some service to biologists is afforded by the question, what is a character? So in some ways, we'll see ourselves as cashing in on something that Hull identified many years ago as a possibly fruitful avenue. So the idea we advanced in the basic model we're putting forward, character identity mechanisms. Character identity is not just a morphological fact, but also something controlled and explained by distinctive causal mechanisms operating in development. Those have consequences for evolutionary persistence and traceability, which is relevant to doing comparative evolutionary work. There's a kind of developmental and evolutionary individuality. Character identity is contrasted with mere similarity between characters and with character states, so that you can have a character like a wing, character state like the size or the shape of the wing. The framework is intended as an abstract model that includes previously studied identity mechanisms as particular realizations while offering a richer and more general account of character identity and homology. and in particular, a way to generalize across different levels of organization, recognizing different mechanisms, different components that also will be relevant to traceability in comparative work. So this is just a snapshot of some of that kind of work. The different types of activities that you might want to pay attention to in mechanisms, different aspects of mechanisms that you would want to trace at different levels of organization. And at those different levels of organization, you might get very different kinds of mechanisms that are accounting for identity. In terms of CHIMs, they have a particular causal profile. I don't have time to go into this in detail, but by characterizing this, we can actually have a phenomenology that can be looked for in empirical work in terms of these four characteristics: actively modular, complex organization, interdependent, causally non-redundant. That particular causal profile actually is something then that you can look for and see a particular kind of signature that is a sort of hourglass behavior where a particular kind of mechanism is refractory to change compared with those inputs that trigger it to initiate and those outputs that it's capable of producing. And this was something that was motivated by an empirical puzzle. For those who have been tracking what's been going on in Evo-Devo, we know that developmental signals and underlying mechanisms can be variable, more variable than we even thought was possible. And yet we also know some of them are deeply the same over long periods of time. How do you reconcile variability and conservation of this kind? Our model is intended as a way of trying to have an intermediate position there. I'm going to skip this one. This is just a criticism of a concept that's currently operative that we think our account does. But let me just highlight this last point, which is elaborating serial homology. Part of what we're doing in developing new theory is actually introducing new terminology that can potentially give us finer discriminations in comparative evolutionary biology that we didn't have available before. Okay, so let me then come to my last case study with my last few minutes here, mutation and evolution. This is some recent work with Adi Livnat at the University of Haifa. So in this recent paper, we distinguished two conceptions of random mutation. One, the familiar that many of us hear all the time: mutation is random if it's not more likely to arise in an environment where it's beneficial. The assumption here is that you're not going to have a change in probability that is relevant to an exposure during the lifetime of the organism. And therefore, if something is a non-random mutation, it's non-random because of a direct response to the organism in its immediate environment. You can actually read that as not Lamarckian, right? And that's kind of the way that's understood by many biologists. The second notion is a mutation whose cause is accidental. Causes are extraneous to the organism's biology. They don't bear any special connection to the phenotype, and therefore their effects are also accidental. No meaningful relationship between the causes of mutations and their evolutionary consequences. Now, once you distinguish two different notions of random, it actually gives you a possibility space to see more things than you could see before. And in this case, what's important is this bottom right category, a not Lamarckian but not accidental mutation, whether that's possible. And part of the motivation for making this distinction was recent empirical work by Adi Livnat's group, where they've been measuring mutation at a high level of accuracy. They've applied it to the human beta globin gene at a 6 base pair region kind of fine-grained level, and identified a mutation rate variation that was far higher than had previously been detected. And in fact, the malaria-resistant mutation originated de novo more frequently, where it provides malaria protection compared to other hemoglobin loci where it does not, and in sub-Saharan Africans who have experienced exposure to malaria for many generations compared to Northern Europeans who have not. And so many of you will recognize this goes against actually the standard story about malarial resistance and the behavior of these alleles in most textbooks. There are other mechanisms that you can pay attention to besides that. This is another one that's in other work. It's called a fused mechanism of gene fusion. which just gives you a way of understanding how particular kinds of mutation might be affected by the behavior of the genome itself. Genes that are used together tend to be fused together. But what these things let us talk about are this new category, which we call complex condition mutations, because the probability origination rate of a specific heritable change can be dependent on that prior information in the genome as a system that has accumulated over many generations under selection pressures, and therefore tie together causes and consequences of mutations in a biologically meaningful manner. And therefore, a mutation can be non-accidental in a manner that is critical for adaptive evolution and yet non-Lamarckian. And so we just name them complex condition mutations to try and capture the sense in which they are separate. And here, just to represent that, is the way it looks. And I know that that was fast. And I know you're probably thinking, wow, but since they're all published papers, people can go back and look at them later. So let me just quickly close. What I just tried to do was show you from 3 areas of my own work, ways in which I exemplify philosophy of biology and interdisciplinary key, which is very much what I think Egenis has done in part over many years, large-scale grant projects, cultivating intellectual community, engaging in individual collaborations. And I think they are an appropriate way to honor the distinctive intellectual legacy of John and Egenis at the University of Exeter.